Good morning, everyone, and welcome to Virtual Grand Rounds. This morning, we're extremely fortunate to be hearing from Dr. Jeremy Luban. Dr. Luban is a physician scientist who studies deadly human pathogens, including HIV-1, Ebola virus, and now SARS-CoV-2. He earned his medical degree from Columbia University here in New York City, followed by internal medicine residency and infectious diseases fellowship, also at Columbia. In his academic career since, he has held endowed professorships at Columbia, where he directed the Columbia Rockefeller Center for AIDS Research at the University of Geneva in Switzerland, and now holds the Freelander Chair in AIDS Research at the University of Massachusetts Medical School, where he serves as Professor of Molecular Medicine and Biochemistry and Molecular Pharmacology. He has been elected to be the American Society for Clinical Investigation, the Association of American Physicians, and is a Fellow of the American Academy of Microbiology. For the past 30 years, his research has been continuously funded by the NIH, including Merit and Avant-Garde Awards. He has served as primary mentor for 20 PhD students, 28 postdocs, and 38 undergrad or master's degree students, on the editorial boards of 4 virology journals, and on grant review and scientific advisory boards for the NIH, the American Cancer Society, the Foundation for AIDS Research, and the Stavros Niarchos Foundation, Wellcome Trust, and the European Research Council. A very warm welcome to Dr. Jeremy Luban.
Thank you, Dr. Mustafa and Dr. Thomas and everyone else for joining us this morning. It's an incredible pleasure to be here with you. I'm going to share my screen if I can succeed in doing that. We're way ahead of the game. Let's see. Okay, and I'm going to hide all of this extra stuff. Give me 1 second. Okay, almost there. All right. So as Dr. Mustafa mentioned, for most of my career, I've been working on a lentivirus, HIV-1. It causes a long, slow disease. And in more recent years, we've turned our focus towards acute viruses, including Ebola and SARS coronavirus 2. And in the course of this talk, I would like to give you a sense of how we've been thinking about these outbreaks. There are some common themes that come up, and I'm just going to jump right in it. So I need to point out that I have no disclosures to make that are relevant to this particular talk. So any day when you open up the New York Times, you see curves like this. This is a death curve from the United Kingdom. It shows you 3 waves over the course of about a year and a half, 2 years. And one interesting thing about this is this was not recent, but this was over 100 years ago. This is what was reported in the records from the influenza outbreak of 1918 and 1919. And I think it's very interesting to think about that outbreak in the context of our current SARS-CoV-2 invasion. Remember, at the time, there was no PCR, there was no deep sequencing. We didn't even really know that nucleic acids were the genetic material. It's interesting to consider that at that time, the leading hypothesis about the etiology was that this was a bacterial infection. And it wasn't really until the 1930s that methods were developed for culturing mammalian cells that would allow you to cultivate viruses and propagate them in the lab. So it was only in the 1930s that the influenza virus was, that people could really start working on it. There are many questions about this outbreak that are relevant to our current outbreak. One wonders where this virus came from. There were these 3 peaks. One wonders what caused these 3 waves. Were these different viruses? Was one a virus and one a bacterium? Are these different variants of concern of the influenza virus. We only have sequence from one of these peaks. Miraculously, it was recovered. And folks there at Mount Sinai, in fact, were instrumental in characterizing the variant of influenza that was actually discovered from this archived material. There are some interesting sources of information. I've put one review down on the left there, and a book called The Great Influenza by John Barry is a reasonable attempt to make sense out of that outbreak. More recently, kind of an interesting contrast is made by the Ebola virus disease epidemic of 2013 to 2016. It was a surprise in that it occurred in a region that had not previously been hit by Ebola, the west coast of Africa here. It began in Guinea and then spread to Sierra Leone and Liberia, as well as several other countries, but this was the epicenter of the outbreak. There were on the order of 30,000 cases, many, many deaths. I would say if you're really interested in learning more about this, there's a very extensively documented book that just came out by Paul Farmer that I would recommend to learn about it. So in contrast to the influenza outbreak of 1918, during this outbreak, this Ebola outbreak was one of the first where sequencing was applied in near real time to characterize the viruses that were being pulled out of people. And over the course of the outbreak, there were many single nucleotide mutations or variants that were found throughout the genome. And most of these were quite rare, perhaps only seen once or a few times, and never really amounted to much during the course of the outbreak. It's also interesting to consider that out of all of these mutations, which were spread out over this 18,000 nucleotide genome, there were only 2 non-synonymous mutations that achieved any kind of prevalence of note. And one of them attracted our attention, which was this mutation in the glycoprotein, which is the protein on the outside of the virus that the virus, this envelope virus, uses to fuse its membrane with target cells. So several groups were sequencing virus out of patient samples during the outbreak. And there are international efforts that most of you've probably heard about now from as a result of the SARS-CoV-2 outbreak, international efforts to collate these sequences and to make sense of them. This nextstrain.org website is one that is very user-friendly and allows you to visualize outbreaks in time, going from left to right. And this is a snapshot taken from it where we focused specifically on this variant residue, the amino acid 82 in this aqua color, and then this 82V variant that appeared. It appeared early in the outbreak, but then it dominated the outbreak. If you look in Guinea, this is a different kind of color coding over time. The original virus is in black. You can see kind of scattered occurrence early in the outbreak of the alanine 82V variant. And then it really dominates in Guinea. And that's the only virus that then spread to Liberia and Sierra Leone. Not to mention, this is the form of the virus that spread to Nigeria, Mali, Senegal, and even to Texas in the United States. And a big question that arose then that has come up again and again during the SARS-CoV-2 outbreak is whether this alanine 82V was just a coincidence. That is, maybe it's just a mutation that appeared and happened to be at the right place at the right time to spread to other countries and expand the outbreak. We attempted to look at metadata, clinical metadata from the outbreak to compare the alanine 82 to the alanine 82V in terms of clinical outcome. And unfortunately, because of the chaos, it was extremely hard to make any kind of comparison. We wondered, is this virus more transmissible? Was it just a result of a coincidental transmission bottleneck, as I just mentioned before, as a possibility? We tried to see if the viral loads were altered, and there seemed to be a trend towards a higher viral load with the valine 82, though it was not statistically significant. There was a statistically significant increase in mortality with this variant, but again, all of this data is confounded by multiple variables, including temporal factors and geographic factors. So it was really not ideal to pin down this kind of thing. So at the time, as I had been, I was working on HIV, and we were studying a totally unrelated question, and really by accident started looking at the effect of these mutations, the alanine 82V mutation, on the ability of virus particles to infect susceptible host cells. And this is a schematic showing the kind of assay we would do, where we would express structural proteins, GAG and POL from HIV, for example, a packageable reporter gene, GFP in this case, and then we can put in trans the glycoprotein from the relevant Ebola virus. And we had done this for other reasons, but Ted Deal, who was the postdoc doing this work, noticed that the alanine 82V mutation in an assay like this was clearly more infectious or better able to enter a target cell than the ancestral alanine form. And we and others have performed a number of other kind of surrogate assays like this. This is an assay where we produce virion particles with the main structural protein or proteins from the Ebola virus. And in similar assays, we saw increased infectivity, including much more complex assays where we put in all of the viral genes. I'll come back to the question of the significance of this mutation in the context of the full virus in a minute, but I want to go forward with some of the experiments that we did going forward. I also want to take a minute to remind you that these glycoproteins are extremely complicated machines that have to assemble into a stable virion particle. This is showing you a kind of schematic structure for the Ebola glycoprotein. The same kind of model could be used to look at the SARS coronavirus 2 spike protein. It's a trimer. It has 3 identical protomers that assemble into this fusion machine. In all cases, there seem to be common pathways by which these trimers promote membrane fusion. And one of the things that often has to happen, and this is particularly true for SARS coronavirus 2, the relevant receptor binding domain has to undergo a conformational change so that it can access the receptor, in this case, ACE2. And this receptor binding often induces an extreme conformational change. There often are proteolytic events that have to remove critical segments. And that then allows the fusion machinery, which is buried underneath, to promote fusion. So the point I want to make here is that there are many mechanisms by which a mutation might increase infectivity. A lot of focus in the news over the past year has been on mutations affecting the binding strength to the receptor, but mutations can have all kinds of other effects that would increase the effective infectivity of a virus particle. So going back to Ebola, we were curious how this very subtle mutation, alanine to valine, in the glycoprotein could increase infectivity. It is located on the molecule near the part of the glycoprotein that binds to NPC1, the main receptor for Ebola virus, but it points away from the receptor, and it wasn't obvious to us how this would increase infectivity. In fact, the alanine 82V interacts with this very dense pocket of interactions involving all of these amino acids internally within the glycoprotein. So we wondered whether the alanine 82 might destabilize these interactions and somehow make the molecule more fusogenic. So maybe it doesn't change binding affinity to the receptor, but maybe it makes the molecule more ready to pop and fuse with the target cell. And so to test this, we generated a series of mutations. This is showing you a model for the effect of the alanine to valine mutation. It takes this small alanine shown here, this little nubbin here, and makes this bigger valine R group here, which would create clashes in this very dense pocket of interactions. And we had shown, as I showed you before, that this mutation would make the virus more infectious. It turns out that anything that makes this area that creates clashes among these amino acids, like a bigger isoleucine, for example, makes the virus even more infectious. Whereas a glycine, which is smaller than alanine, actually makes the virus less infectious. And this is true of a number of mutations. We have about 10 mutations that we predicted would increase the size of this R group on this residue, and all of them increase the infectivity, consistent with this model that destabilization of this pocket is what increases infectivity. And in fact, none of these mutations affect the binding strength. This is a binding assay. What they do though, is make the molecules less stable. And this is an experiment where we heat up the molecule. You can see on the x-axis increasing temperature. And compared to the founder virus, the so-called wild type here, alanine 82, all of these mutations make the molecule fall apart more readily at lower temperatures, which is consistent with this molecule being less stable. We've worked with James Monroe, a colleague of mine here at UMass, to model the effects of the alanine 82V on the protomer. I'm showing you two protomers here in his model, and he can look at the energetics, the positions of the atoms within the molecule, and the effect of this alanine 82V mutation on the critical fusion machinery, which is shown in this kind of aqua-colored loop here. And in fact, when he models alanine 82V, what he sees is that the biggest effects of this mutation are transmitted to the atoms that compose the amino acids in this fusion machinery. And so what we think based on these kinds of biochemical and genetic experiments is that the alanine 82V mutation is somehow destabilizing this protomer so that it's more ready to fuse. Perhaps what it does is it makes the protomers more splayed out and allows the fusion machinery more ready access. We have been working on solving a cryo-EM structure of this protomer, and our preliminary data is consistent with that, but that is still a work in progress. Now, there have been a couple of experiments that have looked at the effect of the alanine 82V mutation on full Ebola virus, but there are a number of problems with those experiments. And there are technical difficulties that it's not readily obvious how to overcome these. So first of all, working with full infectious Ebola, you have to work in a BSL-4. It's very difficult. And the stocks of the viruses that are used there have to be expanded and propagated in tissue culture. And it's been noted by several groups that this passage in tissue culture elicits mutations that have never been seen in people, including this T544I mutation, which in our hands makes our pseudotypes, this assay I showed you before, much more infectious even than the alanine 82V. And so the experiments that have been published with full virus looking at the effects of alanine 82V whether they're in tissue culture or in monkey challenges, in macaques, we've looked at the sequences of these stocks of virus, and they all are densely packed with this mutation. So it's been very difficult to get at this question. Another issue is that this alanine 82V seems to be a human-specific adaptation, or at least a primate-specific adaptation. So the increased infectivity that I told you about, we see on cells from primates, humans, chimps, African green monkeys, for example, but it's not so evident on mice or carnivores, dogs, cats, or in bats. And so experiments that have utilized mouse models and some other animal models, I think, are complicated by this as well. So going forward, this is a big hurdle to pinpointing the importance of this, at least in an animal model or a tissue culture model. But I think the real significance for us at this point is that this mutation tells us a lot about what the virus tends to do, the kinds of selective pressures that are applied to it when it spreads person to person in an outbreak, like what was seen in West Africa. Now, the title of my talk was Deja Vu all over again, and this is the first deja vu, not the second, which I'll talk about in a minute. There was the second biggest Ebola virus outbreak just ended last June, though it's reared its head again much more recently, was in the news, if you happen to notice underneath the SARS-CoV-2 outbreak. This one occurred in the Democratic Republic of the Congo, in a very difficult part of the country that is war-torn for decades. There were many cases and many deaths, and working with JJ Mayembe, who's in Kinshasa, his large group there has been doing sequencing with assistance from folks in the US and in the UK. And the sequences that they've put together so far, interestingly, there seems to be a different mutation in the glycoprotein, this valine 75 to alanine, that has indeed taken over the outbreak. So it wasn't present in the ancestral virus, but towards the middle and the end, it was the only form of the virus that was seen. We're still trying to fill in the gaps in this sequencing. And when we test this virus in our pseudotype assay, either on this cell line U2OS cells or on primary human monocytes, each of these dots is a different human blood donor, each in triplicate. What we see is that this mutation also is more infectious. And so this mutation, we're taking apart the same way we did the alanine 82V mutation. We will endeavor to solve its structure, and we expect that it may have a very similar mechanism in terms of how it promotes infectivity. So I want to go back in time again briefly to tell you about how we first learned about coronaviruses. This book, Cold Wars, is another book I would recommend to you if you can get a hold of it. It's out of print, but it is still possible to find copies online. It details the history of the Common Cold Unit in Salisbury, UK, which was created after World War II. The Brits were quite aware of the importance of viral infections for the security of their nation. Viruses can devastate troops in wartime. And this was initially a military facility, and then it became just more generally a research unit where some of the really important findings about common cold viruses, ultimately rhinoviruses, flu, were characterized in terms of the clinical course. They literally brought volunteers. These are the first volunteers who were brought in. They were challenged with extracts from snot, in effect, and this is someone quantitating the disease by measuring the weight of the secretions that people were producing. Now, at some point, the director of the unit, David Terrell, who was one of the authors of that book, had an isolate that was able to cause disease in people, but they were having trouble cultivating it and trouble characterizing it. And he sent it to an investigator named June Almeida, who was in London at the time. This is a picture of her in Canada. She's from Glasgow. She wanted to go to university, but couldn't afford it, went and became a lab technician, and ultimately became the world's expert at the time as an electron microscopist characterizing viruses. So David Terrell brought her a sample. As he said, we were not too hopeful, but felt it was worth a try. And in effect, what she saw were particles that look like this. This is not her exact image, but you can look back in the literature and find those. She recognized this as something unusual that she had seen before in animal infections. And it turns out her paper describing these particles was initially rejected. I think it took her 2 years or so to publish them. It was thought that they were just sloppy EMs of influenza virus. We now know that this is a common cold coronavirus that all of us have been exposed to. So there are 4 such viruses that circulate. And in the past couple of decades, there have been these horrendous coronavirus exceptions, the original SARS, MERS, and now SARS coronavirus 2. SARS coronavirus 2, like the other coronaviruses, is extraordinary for having the largest genome among the RNA viruses. Its size is so big that it's beyond the theoretical limit of viability for an RNA virus because the replication machinery generally is very error prone. And if too many errors appear, the virus cannot replicate. It turns out that coronaviruses have a proofreading function, this NSP14 exonuclease that is thought to minimize the errors during the virus replication. And as a result, these viruses as a class have the reputation of having relatively low rates of mutations. And that's been determined experimentally in the lab and in other outbreaks. And in fact, in the beginning of the COVID pandemic, there was the sense and there was the opinion of many virologists that this virus really didn't pose a challenge in terms of eliciting new variants over the course of the outbreak. There have been tens of thousands of single nucleotide polymorphisms seen. And again, this is perhaps the deja vu all over again. There really are only 2 non-synonymous single nucleotide polymorphisms that rose to high frequency over, let's say, the first 6 months of the outbreak. And this shows that back in February, we noticed there was an amino acid change in the glycoprotein, the spike protein, changing the aspartate 614 to a glycine. It appeared relatively early, again, as the virus was spreading person to person. It then started to dominate, and by June, it was really the only form that was seen around the planet. This is a snapshot of the phylogeny taken this January. So we wondered again, is this mutation more infectious? And this is Lanya, a postdoc working with me. He set up these pseudotype assays that I showed you before, where he tests the ability of the spike protein variants to enter susceptible target cells. And he's comparing the ancestral form, the D614, with the D614G. So these are isogenic, except for that one mutation. And these are several different contexts where these were tested. These are callo-3 lung cells, which of the cell lines, in our opinion and the opinion of a number of groups, seems to be the cell line that most closely reflects what actually happens when the virus enters real lung cells. This is a colon cancer cell line that is also ACE2 positive and has the TMPRSS2 molecule that allows the virus to enter the way it does in real lung cells. And then these are two artificial systems. These cells, Do9-3 cells or SUPT1 cells, they're very different from each other. Neither is susceptible to coronavirus infection, but we delivered the receptors genetically, the ACE2 and the TMPRSS2 protease that are important for viral entry into lung cells, into these cells, and render them susceptible. And when they become susceptible, this mutation then increases infectivity. So I would say these initial studies, which we reported first in bioRxiv and then published in a peer-reviewed publication, very similar results were reported by at least 4 other labs at the time. And I would say there was enormous pushback from the community. Many people thought that it was ridiculous what we were talking about. And we were told that this virus doesn't mutate in any significant way and that these couldn't possibly be important. And I think it's become quite clear now that this is not true, and that while SARS-CoV-2 may not mutate at the same rate as influenza or HIV, it certainly is capable of acquiring mutations that are adaptations in some way that have been selected for some property that may make it a problem for us in the control of the pandemic. So since then, there's been confirmation in the context of full reverse genetic SARS-CoV-2 virus. These are experiments done in BSL-3 labs. And the mutation has been shown to increase replication as well as transmission of the virus. These are the 3 main labs that have been doing these kind of reverse genetic experiments. The ultimate experiment, for example, is to challenge an animal, a hamster or a ferret, for example, with the virus, and then put it in proximity to uninfected animals. And in those kinds of experiments, it was shown that the virus was more transmissible. Many studies have shown increased viral load in people who are infected with SARS-CoV-2 if they have this variant. Luckily, there have been no reports of increased pathology, increased case fatality rates with this mutation. And attempts have been made to try and show increased transmission rates in humans, and it's an extremely difficult thing to demonstrate because, as was the case in the UK, the populations that had the different forms of the virus were very different in terms of their demographics. So, for example, the D614G originally arrived in the UK in young people returning from the Alps who had been skiing, very different population from, say, middle-aged businessmen coming back from China. So a big question in the news and on all of our minds is, where did these viruses originate? This is a phylogenetic tree showing you the original SARS coronavirus, which is by all intents and purposes now extinct, but it clearly originated in bats. If you go back in time, maybe it came to humans through an intermediate host like the civet. Similarly, SARS coronavirus 2 has its origins, its most closest relatives in bats. There's some question about whether there's an intermediate host like a pangolin, but the bottom line is that the most proximal reservoir is still unknown. We performed these kind of pseudotype assays on target cells that had ACE2 orthologs from different species. So this is an experiment with the human ACE2 looking at the effect of D614G. It's more infectious on human. Similarly, in this horseshoe bat, ACE2, the virus is more infectious. Same is true in the pangolin, the potential intermediate host. And we see that this mutation is more infectious in our pseudotype assays using any permissive ACE2 ortholog that we've looked at. And this raises a very interesting question, which is, why was D614G never seen before in other coronaviruses? And one potential explanation is that SARS-CoV-2 has a polybasic protease cleavage site encoded within it that is relatively unique, at least in this subclass of coronaviruses. And this is showing you this polybasic site. It's cleaved by, among others, the furin protease when the virus assembles. And if we mutate the furin site, what we see, at least in these very artificial entry assays, cleavage of the furin site makes the virus more infectious as compared to the virus that has the furin site in these pseudotype assays. And in that context, D614G no longer has an effect. I should point out that the furin cleavage site is critical for the infectivity of SARS coronavirus 2 in the context of the full virus, and is clearly important in the pandemic. But in this artificial assay, at least on a kind of biochemical level, there's an interaction, we think, between 614G and this furin cleavage site. One question that we had is whether 614G would increase the binding to ACE2, and this is just showing you a whole bunch of data, the conclusion of which is that the mutation does not increase the binding strength. If anything, it increases the off rate, which in effect translates into a decrease in binding strength. So again, I mentioned before, mutations can increase infectivity via many mechanisms. We don't think D614G increases infectivity by increasing binding to the receptor. Good news about that mutation is that it does not appear to be resistant to monoclonal antibodies. As shown here, these are experiments from our collaborators at Regeneron with their monoclonals, or to convalescent plasma, or from plasma from people who have been immunized with any of the SARS-CoV-2 vaccines. So that is one little piece of good news here. Working with Quan Chen, who's a new assistant professor in my department, we produced SARS-CoV-2 protein, spike protein, the D form, the original form, and the G form, the proteins in this expression system, which has been used locally by Professor Kramer at Mount Sinai. These two proteins were very well behaved. They were very readily imaged by cryo-EM. These are photographs of individual protomers. These are trimeric particles of these proteins, and you can see different orientations. These little white dots here, for example, are alpha helices on end. It's amazing what one can do with technology these days. And the most impressive finding that we had when we looked at these particles was that the molecule was splayed open, and we think we know the molecular basis for that at the atomic level. And a consequence was that the receptor binding domain was much more open, pointing upwards. And I'll show you a movie of what that looks like in the mutation. And we think that the fact that the protomers are much more open in D614G than in D614 makes this molecule much more ready to fuse and promote entry. There are other effects, including stability of the complex on virion particles, but this was something that came out of our analysis of the structure that we think is very interesting and maybe analogous to what we saw with the Ebola virus. So this is a model of the protomer. You can see each protomer has a different color. And the RBD is shown lifting up and pointing towards ACE2 in the current models of how we think this molecule works. So we think D614G has a more open conformation. We think it's more primed to bind ACE2 and fuse, even though it doesn't bind more tightly. It's clear that these viruses are more infectious and more transmissible. They're not resistant to neutralization. And one of the messages that came out of these experiments, which were done by last summer, was that there was a very important need to monitor the virus as it spread around the planet. And unfortunately, there was a pretty big lag in sequencing last summer, even as the virus was continuing to spread. And we now know that this virus is capable of eliciting much more concerning mutations than this. So many of us have our email boxes or phones ringing off the hook all the time. Reporters are constantly interested in the new variants, and there are reports coming out all the time. It's been extremely hard for everyone to keep track of them. These are the big three, the B117, which was first described in the UK. We don't know for sure that it originated there. It's just that the UK has done by far and away the best job of any country at sequencing viruses and linking those sequences to metadata, either epidemiologic or clinical. P1 was first reported from Japan and people who had returned from Brazil. And the 351, B1351 variant that was first described in South Africa, which is done, South Africa has done an extraordinary job of monitoring the outbreak and looking at the viral sequences in those. The United States overall has done a terrible job. This is an article from back in December showing our position in terms of the frequency of sequences that we were generating in terms of monitoring the outbreak. Things are slowly improving. There's an attempt to coordinate efforts across the United States to get representative sampling of the viruses as they spread. The UK has a complex system of sequencing where they sequence up to 10% of all of the viruses infecting people. It's thought that 5% is kind of the minimal target that we'd like to achieve. We've brought this 0.3% up a little bit since December, but we still have a ways to go in terms of really knowing what the denominator is of the different variants. So what makes a SARS-CoV-2 variant of concern? Again, these are the big three, and these are the amino acid changes that are seen just in the spike. And that's an important point to remember. Much of the virus is not being considered in this discourse. What was really shocking about B117 when it first appeared is that it was the sudden appearance of a cluster of mutations, many mutations. And up until then, the international sequencing effort was reporting in the order of 2 mutations per month that were getting fixed within the virus population. After the appearance of this virus, all of a sudden that number jumped up tenfold. And so over the past fall, we've seen viruses with large collections of mutations. All of these, each of these three, we believe enhance transmission. There's pretty good evidence for that. The B117 was very important because it eluded some of the diagnostic tests that once that was realized, it became a kind of a useful trick to identify the virus without full sequencing. But it's something that we have to think about going forward. We have to be sure that any diagnostic tests are still functional. I think B117, in my view, has been quite convincingly shown to be more pathogenic. That is, people who get infected with this virus have a higher probability of getting sick or getting hospitalized or dying. And there's some concern that maybe there's a higher frequency of symptomatic infection in younger people as well. There's a constant effort to assess the potential of these viruses to reinfect people who've previously been infected, and this is a kind of a moving target. It seems quite clear that that these viruses as a group are capable of infecting people who've been previously infected. Maybe B117 has the least concern in that front. And of course, going forward, everyone's concerned about whether these viruses will escape from the protective immunity conferred by vaccination. This is kind of a vignette from the Amazonas in Brazil, where I'd say information is extremely incomplete, and it's just a vignette, and it's not clear really what the significance of this is. There was a report in Science that came out saying that by October, 70% of the population had been infected with the virus from the first wave, and therefore that the population might have achieved some sort of herd immunity. And then all of a sudden in December, there was an explosion of cases. The whole infrastructure was overridden by COVID. People couldn't get oxygen, and that corresponded temporally with the first detection of P1. Now, we don't really know when it first appeared because the sampling is inexact. We don't really know what the herd immunity was, but there was the suggestion that this virus was capable of infecting people. There clearly are cases of people who had been infected, who did get infected, and it kind of still remains to be determined how facile this virus is at escaping the convalescent immunity from prior infection. I would say most of the experiments that have been reported so far indicate that vaccination appears to provide better protection than prior infection. That may be because the virus is really adapted to avoid the immune response. I think there is hope that even with these viruses, in the majority of cases at least, vaccination will make a dent in the outbreak. So where is kind of a last question I want to address, where do these variants of concern come from? Why all of a sudden was there a whole cluster of mutations? And the most important clue we have is rare case reports of persistent infection within individuals. So for example, my colleagues here in Boston reported an individual who was on immunosuppressive therapy for an autoimmune disease. They had a very long course where they were infected with SARS-CoV-2 over a period of months. And when they sequenced that virus, many mutations like those in the B117 variant were discovered. And one has the sense that if the virus is capable of replicating intra-host and doesn't have to jump to another person, it may have time to sample more genetic space, more mutations, and come up with combinations of mutations that allow it to survive, be more infectious, but also spread through the air. There's also the fact that the initial outbreak was in a naive population, and it's possible that some of these variants that are appearing are being selected by the convalescent immune response in the people who were infected last spring. And finally, I think we have evidence that D614G may be acting epistatically. That is, it may be permitting other mutations to exist that otherwise would not be viable. And this is just one experiment that supports this. We have a number that we're looking at now along these lines. If we look in the background of the original virus, D614G increases infectivity here on the order of 5-fold. When we look at B117, at the effect of this D614G mutation, which is always present in B117, we find that the virus is extremely dependent on this mutation. And if we revert the D614G to the D614, the virus is quite sick. And we think this may be a general pattern going forward. So going forward, it's really important that we continue to interrogate the sequence databases and look for scary mutations based on their prevalence, their likely transmissibility if we have kinetic data, looking at outbreak clusters and looking at their novelty. Currently, there are more than 1 million sequences from around the world in the GSED database. We need to continue to engage in screens in the lab to predict new variants. There have been some very important screens, both biochemical and functional. For example, Regeneron selected the monoclonal antibodies that they put in their cocktail by looking for the ability of the virus to escape from monoclonals. They put together a combination that, at least in the lab, the virus could not escape from. And we need to monitor specific cases, for example, immunocompromised people, where the selective pressure on the virus may be altered and the virus may have a chance to grow and mutate. And of course, the real big question now everyone is focusing on is people who've been vaccinated who become infected. What accounts for that? And so far, I'd say the biggest series I've seen, which comes from Boston University Medical Center, their impression at this point is that the major determinant is actually host factors and not so much the virus, but that remains to be seen. We need to set up systems so we can link any of this epidemiology or molecular biology to clinical metadata. We need to continue to monitor in better assays the phenotypes of these mutations, as well as mutations outside of spike, especially if we develop drugs targeting other genes, and the immunology and biochemistry and animal models need to be maintained. And so I think I better stop at this point. My group is shown in this artsy black and white here on the left. These are the people who continued working, Yitao, Thomas, Alanya, and Anne, primarily throughout the pandemic, working on the variants as they've appeared. And we've been working extensively with Quan on the biochemistry and structural assessment of the variants as they appear. This is an ongoing effort. And the epidemiology has been critical for our efforts working with Party Sabetti's group at the Broad. And so I'll stop here and see if anyone has any questions.
Great, thank you. Thank you very much. That was very interesting. So let's see if anyone has some questions that they can put in the chat. There goes one, John.
So the question is, will we all need annual boosters? And I think if you ask experts, the fact is none of them can answer that question. I would say we have two kind of precedents that we can use to think about what may happen going forward. So one precedent is the seasonal coronaviruses. There are 4 of them. We've all been infected with probably all 4 of them by the time we're 18. They seem to cause modest disease in young people, common cold type symptoms usually. And perhaps once everyone's been exposed to SARS coronavirus 2, the so-called herd immunity at some level, the virus will be relegated to that kind of a state. That is, it will be a less serious concern than it currently is. I think that is indeed one possible outcome of this outbreak. There are no vaccines against the other coronaviruses, and indeed it may turn out that once we get through this this pandemic, there won't be a need for vaccination going forward. The other extreme possibility is what happened with SARS, the original SARS coronavirus #1. It was significantly more deadly than the current SARS coronavirus 2, and it wiped itself out because of that. So it was readily apparent if someone was infected, there was not an asymptomatic state, and it was relatively easy to control that outbreak. It could be that SARS coronavirus 2 is somewhere in the middle, and I think that's what a lot of people are thinking. That is, well, you may get some sort of immunity if you've been infected with the virus. This virus is particularly wily, and the immune response that it elicits is maybe not as good as you would like. Clearly, the vaccination response from a number of studies looks to be better than the convalescent immune response in terms of the quality and perhaps ultimately in terms of its duration. So I think it's conceivable that if people are immunized with a vaccine, if we could possibly get to the point where we vaccinate people, and remember, most of the world has not even started vaccinating populations, we might be able to get to the state where there's enough kind of baseline immunity, and maybe if younger people get infected, their immune response is more stable. But I think there's a good chance that there may be a need for boosters. Maybe the immune response from the vaccine will be strong enough so that boosters won't be needed. The other question in everyone's mind is whether the variants are going to escape from the immunity. I have to say, it looks like even the current vaccines that are being given based on the original virus are quite effective against the scary variants that are being transmitted. There are reductions in the efficacy in specific parameters like the neutralization titer. Some of the viruses like the B.1.351 first reported from South Africa, many convalescent samples are completely unable to neutralize. There are efforts now to develop a 351 based vaccine, and those are being worked up. And I imagine going forward that will, in the not too distant future, will be combined with the original vaccine or replace it. The other piece of good news is that the immunity against 351 does work against the original virus. So it seems that it may be that just getting vaccinated against one of these newer variants will protect you against all the things that are coming. The big unknown is what lies ahead. And it's not clear whether the virus has additional pathways it can explore to get to the receptor, to get into cells to cause disease in the face of the immunity that's been established against the current variants. And there again, if you poll all the experts in the field, they all have different opinions. Some think the virus has an infinite capacity to explore genetic space. I'm of the opinion that there's only so much that the virus can do. It really, in order to transmit, in order to cause disease, it has to be adapted pretty tightly to very narrow parameters in our airway. So I am an optimist going forward. I think the real concern, though, is that we need to get the vaccine to more people, whether that be people who don't want to get the vaccine in the US or people in countries where the vaccines are just not available. Maybe that was too much, but that's my 4 cents.
That was great. And I think on the interest of time, we'll have to bring it to a conclusion, but that was a great ending. And I like the fact that you're an optimist with your expertise. So thank you very much for spending the morning with us, Dr. Luban. It was really quite interesting. It was great to have you here. We appreciate it.
Thank you so much. Thanks, everyone. And feel free to contact me if you have questions. Be happy to reply.
Great. Have a good day, everybody. Thank you.